Introduction. Hey, today I am very happy because my aunt gave birth a baby girl. That's great. But my uncle was not happy because he wanted a baby boy. He was blaming himself for this. But why he was doing that? Actually, a mother is not only responsible for giving birth of a female child, but it depends on the father's gene that actually decides the sex of a child. Let me explain you this mechanism in detail. Objectives. At the end of this lesson, you will be able to understand the mechanism of sex determination, understand mutation, understand pedigree analysis, identify various types of genetic disorders. Sex determination. Chromosomes can be classified into two types. They are sex chromosomes and autosomes. Sex chromosomes, a chromosome involved in determining the sex of an organism, usually designated as X or Y. Autosomes. All the pairs of chromosomes other than the sex chromosomes are called autosomes. Sex determination mechanisms can be classified into three types: exotype, XY type, ZW type. Exotype. In this, males have only one X chromosome, while females have a pair of X chromosomes. Example, grasshopper. XY type. In this, males have only one X and one Y chromosome, while females have a pair of X chromosomes. Example, human being. ZW type. In this, females have only one Z and one W chromosome, while males have a pair of Z chromosomes. Example, birds. Sex determination in humans. Sex determination mechanism in human beings is XY type. Human beings contain 23 pairs of chromosomes, 22 pairs of autosomes, and one pair of sex chromosomes, XX in females and XY in males. During fertilization of an ovum with sperm, two types of zygotes can be formed. Therefore, there are 50% of birth of a male XY or female child XX. Thus, the genetic makeup of the father's sperm is responsible for sex determination and not the mother. Mutations. Variations can be found in progeny due to: number 1, recombinations after crossing over of chromosomes. Number 2, mutations. Mutation. Mutation is a phenomena which results in alteration of DNA sequences and consequently results in changes in the genotype and the phenotype of an organism. Mutagen. Factors that induce mutations are called mutagens. Mutagens can be chemicals such as alkylating agents, radiations such as UV rays, ionizing radiations, etc. Virus. Physical agents. Types of mutations. Mutations can be classified into the following types. Deletion. Deletions remove one or more nucleotides from the DNA. Insertion or duplication. Insertions add one or more extra nucleotides into the DNA. Point mutations. These are the mutations arising due to the change in a single base pair of DNA. Frame shift mutation. In this, one or more bases are inserted or deleted. As a result, the DNA sequences become meaningless. Pedigree analysis. Pedigree analysis. An analysis of traits in several generations of a family is known as pedigree analysis. Pedigree. It is a diagram of family relationships that uses symbols to represent people and lines to represent genetic relationships. Some basic symbols used in a pedigree are: genetic disorders. A genetic disorder is a condition that is caused by an abnormality in individual's DNA. Mendelian disorders. A genetic disease showing a Mendelian pattern of inheritance and caused by a single mutation in the structure of DNA is known as Mendelian disorder. Such a disease trait can be dominant or recessive. Pedigree analysis can be used to determine the transmission of such traits. Mendelian disorders. Some of the most common Mendelian disorders are hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria. Hemophilia. It is a sex-linked recessive disorder that impairs the body's ability to control blood coagulation. It is caused due to the deficiency of certain clotting factors. The transmission of this trait occurs from carrier females to unaffected male progeny. The possibility of a female becoming a hemophilic is extremely rare because mother of such female has to be at least carrier and the father should be hemophilic, unviable in the later stage of life. The following chart shows the transmission of hemophilia. Cystic fibrosis. Cystic fibrosis is a genetic disease caused by deletion of the gene coding for the protein CFTR, which is located on the long arm of chromosome 7. The loss of usually 3 nucleotides results in changes in sequence and conformation of the protein CFTR, which makes it dysfunctional. The protein CFTR controls the transport of chloride ions across the cell surface membrane. It damages the electrolyte balance of the organism and results in change in exocrine glands. The membranes lining the lungs produce abnormally viscous mucus, resulting in frequent infections and breathing difficulties. The disease also leads to enzymatic failure of the pancreas. This in turn causes disorder in the digestion and absorption of food. Long-term retention of secretion in these glands results in fibrosis of the secretory organs such as the liver and pancreas. Secretory dysfunction causes the sweat gland to produce sweat containing increased concentration of sodium chloride. This is referred to as salty sweat. Sickle cell anemia. This is an autosomal recessive trait transmitted from parents to the offspring when both the partners are carrier for the gene. The disease is controlled by a single pair of allele, HBA and HBS, only homozygous individuals, HBS. The defect is caused by the substitution of glutamic acid bivaline at the 6th position of the beta chain of hemoglobin. Thus, the shape of RBCs changes to elongated sickle cell structure. Phenylketonuria. It is an autosomal recessive disorder that occurs due to accumulation of phenylalanine in the brain, resulting in mental retardation. Chromosomal disorders. These are caused due to absence or excess or abnormal arrangement of one or more chromosomes. Such disorders can be aneuploidy. Failure of segregation of chromatids during cell division cycle results in the gain or loss of chromosomes, called aneuploidy. Polyploidy. Polyploidy is a condition that arises when cytokinesis does not occur after telophase, resulting in increase in a whole set of chromosomes. Trisomy. An extra copy of a chromosome is called trisomy. Monosomy. Absence of one chromosome from a pair is called monosomy. Chromosomal disorders. Some common chromosomal disorders in human beings are Down's syndrome. It occurs due to the trisomy of chromosome number 21. Klinefelter's syndrome. It occurs due to the presence of extra copy of X chromosome in human sperms XXY. Turner's syndrome. It occurs due to the absence of X chromosome in human ovum XO. Did you know? Donase Alpha is a drug prepared by co-transformation technique by a US biotechnology firm. It aids in the treatment of cystic fibrosis, a Mendelian genetic disorder. Summary. Let us summarize what we have learnt. Sex determination mechanism is of three types: XO, XY and ZW. Human beings follow XY type of sex determination. Mutation is defined as change in the genetic material. A genetic disease showing a Mendelian pattern of inheritance and caused by a single mutation in the structure of DNA is known as Mendelian disorder. Example: sickle cell anemia, hemophilia, etc. Down syndrome occurs due to trisomy of chromosome 21. Klinefelter's syndrome carries an extra X chromosome, while Turner's syndrome lacks a X chromosome.